Hello, and welcome to Learning Science Daily. In this video, we'll be diving into Part 3 of the chapter, Introduction to Biopharmaceutics and Pharmacokinetics. If you're new here and want to stay updated with daily science content, don't forget to subscribe to the channel and hit the bell icon. When we talk about drug exposure and drug response in pharmacology, we're looking at two key aspects of how a drug works in the body. Drug exposure refers to how much of the drug enters the body, and how it behaves over time. This includes measurements like the highest concentration of the drug in the blood, which is known as C maximum, the lowest, known as C minimum, how long it stays at a steady level, which is known as CSS, and the total amount of drug the body is exposed to, or AUC. These help us understand the drug's availability in the system. Drug response, on the other hand, is about what the drug actually does once it's in the body. This can be as basic as whether it binds to the right receptor, or as complex as whether it leads to improvements in clinical outcomes like lower blood pressure or better heart function. So, in simple terms, exposure is about how much drug the body sees, while response is about how the body reacts to it. Toxicokinetics is the study of how a drug or chemical moves through the body when it is present at harmful or toxic levels, especially during preclinical testing in animals. It applies the same principles as pharmacokinetics, absorption, distribution, metabolism, and excretion, but focuses on understanding exposure during toxicity studies. This helps scientists determine whether the toxic effects seen in animals are relevant to humans, especially when predicting safe dose levels. On the other hand, clinical toxicology deals with how toxic substances, including overdosed drugs, affect the human body. It's important to note that when someone is overdosed, the body may handle the drug very differently than when it is given in safe, therapeutic doses. We'll explore these differences in more detail as we go further. To understand how a drug behaves in the body, we need to measure how much of it is present at different times. This is a key part of pharmacokinetics. These measurements are usually done using biological samples like blood plasma, urine, saliva, or even milk. To do this accurately, we rely on highly sensitive and precise laboratory techniques. The most common methods are chromatography and mass spectrometry. Chromatography helps separate the drug from other substances in the sample that could interfere with the results, while mass spectrometry detects and measures the drug molecules by identifying their unique mass-to-charge ratios. These techniques are carefully validated to ensure the results are reliable, which is essential not only for research but also for monitoring drug levels in patients during treatment. When we collect samples to measure drug concentrations in the body, the method can be either invasive or non-invasive. Invasive sampling involves collecting biological materials that require a needle or surgery, like drawing blood, taking spinal fluid, joint fluid, synovial fluid, or doing a tissue biopsy. These methods can be uncomfortable or carry some risk, so they're used carefully. On the other hand, non-invasive sampling is much simpler and safer. It includes collecting things like urine, saliva, feces, or even expired air, which don't require breaking the skin or entering the body. Non-invasive methods are often preferred when possible, especially for repeated testing or in vulnerable patients. When studying how a drug moves through the body, measuring the drug and its metabolites in the blood is one of the most direct ways to understand its behavior. Blood is made up of cells like red and white blood cells and platelets, as well as proteins such as albumin and globulins. To analyze drugs, scientists usually focus on either serum or plasma rather than whole blood. Serum is the clear fluid you get after blood clots, and is spun down in a centrifuge, while plasma is the liquid part of blood obtained by adding an anticoagulant like heparin before spinning it down, so it doesn't clot. Because of this difference, serum and plasma have different protein contents. Plasma circulates through all the tissues and carries the drug, often bound to proteins. When measuring drug levels in plasma, you can either measure the total amount, which include both bound and unbound to proteins, or just the unbound, active portion after removing proteins. Knowing which type of plasma concentration you are looking at is important because it affects how you interpret the drug's presence and activity in the body. Now is the time for questions and answers. Question number 1. Why are drug concentrations more often measured in plasma rather than whole blood or serum? And the answer is, drug concentrations are more often measured in plasma because plasma contains the liquid portion of blood with anticoagulants that prevent clotting, preserving the drug and its binding to proteins as it circulates in the body, making it a more accurate and consistent sample than whole blood or serum for assessing drug levels. Now, second question, what are the differences between bound drug, unbound drug, total drug, parent drug, and metabolite drug concentrations in the plasma? Here's a simple explanation of these terms. Bound drug concentration is the portion of the drug that is attached to plasma proteins like albumin, and is inactive because it cannot easily enter tissues or have an effect. An unbound drug concentration is the free portion of the drug in plasma that is not attached to proteins, and is active, able to move into tissues and produce effects. The total drug concentration is the sum of both bound and unbound drug concentrations in plasma. The parent drug concentration is the original form of the drug as it was administered before the body breaks it down. And metabolite drug concentration is the substances formed when the parent drug is chemically changed or metabolized by the body, which may be active or inactive. Thanks for watching. If you found this video helpful, don't forget to like, share, and subscribe for more. Drop your questions or thoughts in the comments. I'd love to hear from you. See you in the next video.